Good afternoon. We will get started in just 2 minutes. Good afternoon, and welcome to the August installment of the Faces of Agriculture and USDA Speaker Series, hosted by the USDA Office of Partnerships and Public Engagement. The purpose of this speaker series is to connect students with current USDA researchers, scientists, technicians, engineers, foresters, other STEM-related program managers to learn about their story, career path, and exciting projects and initiatives they may be working on at USDA that serve the public interest, while helping to inspire the next generation of agricultural workforce. In addition to today's speakers, we invite students to visit USDA's Youth and Agriculture website to learn more about internship, scholarship, and other great programs for youth. The website can be found at www.usda.gov/youth. Before we begin, we ask all attendees to place their phones and devices on mute. This event will be recorded, and a copy of the recording will be available to participants by the end of the week. We welcome and encourage all questions for our speakers. If you have questions for our speakers, please place your question in the chat box and we will address all questions at the end. I want to take a moment to thank our speakers and participants for taking time out of your busy day to learn and grow as a community. Without further delay, I would like to introduce our speakers. Dr. Jay Evans is a research leader for the USDA Agricultural Research Service Bee Research Laboratory in Beltsville, Maryland. The research lab is focused on the development of management strategies to help honeybees thrive in the face of disease, chemical stress, and inadequate forage. Lab members are developing and testing new nutritional and anti-disease products and are especially interested in how bees respond to multiple stress factors and the efforts beekeepers might use to help them through these challenges. Current projects involve honeybee immunity, interactions among stress factors, and the development of novel safe controls for mites and viruses. Jay received his undergraduate degree in biology from Princeton University and his PhD in biology from the University of Utah. Mr. Sam Aban is the director of the USDA Agricultural Research Service National Honeybee Disease Service. This service receives over 2,000 honeybee samples annually from beekeepers and inspectors Diagnostics are provided for parasitic mites, gut parasites, and two important bacterial diseases of honeybees, usually within 1 week. This service has been provided by the USDA since the early 1900s, initially from the USDA office in Washington, D.C. Sam has been part of the Bee Research Lab for many years and helps describe the challenges facing honeybees to a wide audience. Prior to joining USDA, he earned his Bachelor's of Science in Biochemistry from Washington Adventist University. Dr. Samuel Ramsey's enduring interest in entomology started over 20 years ago and shows no sign of fading. Having completed his PhD in 2018 with Dr. Dennis Van Engelsdorp's lab at the University of Maryland, College Park, Dr. Ramsey maintained a focus on how insect research can benefit the public through the development of IPM strategies and STEM outreach initiatives. Dr. Ramsey studied entomology at Cornell University as an undergraduate, focusing on predatory parasitic insect behavior. His current work at the USDA ARS Bee Research Lab as an ARISE Fellow focuses on the effects of honeybee parasites on individual and colony level survivorship. Without further ado, I would like to introduce our first speaker, Dr. Jay Evans.
Thank you very much, Rudy, and thanks to all of you for this. It's an honor to speak to students. We were all students and still are in many ways learning what we're doing. That's a good thing about science. You're always learning something new. But I also remember as a student, it was, I had no idea. I certainly didn't know I would work with honeybees as a career. I was probably thinking, something larger and furrier than a honeybee to study. I guess they're furry. But it was really through teachers in science that I really got an interest, at least in knowing that I wanted to do science of some form. And we, on the other hand, in our lab, we have many students coming through working in the summers, some high school students in the fall, and they're really inspiring because they bring new skills and a lot of energy to the group. So it is an honor to talk to all of you, and the three of us will kind of divide up the time. I'm going to give more or less an overview of the laboratory where we work and different projects there. And then Samuel Aban will give a discussion of the diagnostic service that he's running and the history and the nature of that. And then Samuel Ramsey will talk about some really cool stuff with a really bad creature, which is a mite that parasitizes bees. So I will show you a few slides to start. Again, I'm really, let's see, yeah, just checking. There we go, sorry about that. Is that showing up on the screen? Ruby? Can you all see that on the screen?
Yeah, it's on now.
Okay, thank you. Sorry, it took a little minute. I'm getting the ropes here. Okay, so what I'd like to do is talk about our lab's focus and also some of the folks in the lab doing this science. And as you might imagine, we're part of many other scientists at USDA. You can study soybeans, cows, bees, water, soils, everything. And we happen to be a lab that focuses on honeybees because they're important. They help many crops in terms of pollination, and they kind of keep our food more interesting. So this is our team from a couple summers ago, and there are only 11 or 12 of us who are permanent employees, and then we again have lots of students, some from overseas, some from the US, from high school students to graduate students visiting the lab. And so we have a kind of a rotating group of people training and helping with different projects in the lab. And as Ruby had mentioned, our real focus is on managing honeybee disease and honeybee stresses. So we just want to find ways that a colony doesn't have to deal with all of the stress that they might face. And I just have to describe very briefly what happens to colonies when things go badly. You know, individual bees die, sadly, not that uncommonly, right? There's thousands of bees and they only live about a month each one. But what we're really worried about is when the whole colony, which can be maybe, 40, 50,000 bees, when it collapses. And so we want to know why that happens. Sometimes it's a tree, other times it's a disease, and kind of sort out more or less exactly why it's going on and how we can stop it. And here's your first data for this presentation. You don't need to remember the numbers so much, but just bear in mind that many honeybee colonies die each year. It's really hard lately for beekeepers to keep their colonies going. We want to get that rate down to about 15%. This sort of the purple bar at 15%, but we're not getting there yet. We're way above that, and we're trying to fix that. Here's the few reasons that this could happen. I also want to just, since I like science probably too much, I'm going to show you some studies, just overviews of them, so you see kind of the nature of not just what people are discovering, but how the science is done. This is a study on one issue of chemical stress with bees, but what was really interesting about it is what the scientists did, and we're actually doing some work like this in the lab, getting towards this. You can put little tags on each and every bee in a colony, and those tagged bees, when they check in to the hive, you know that bee just came past the door or it went out the door. And so these tags are helping us kind of figure out, which bees are actively foraging, collecting plants, and then sadly, it also tells us when the bee dies because she doesn't show up for the next roll call with the tag. So you get very good data on that. In this particular study, it was sort of bees that were under stress kind of steadily did less well than the other bees. And we see this, whether it's from viruses or the mites that Dr. Ramsey will talk about, or other stresses. The bees that are facing that stress, they don't just fall over dead, on day one, but they steadily die at a higher rate than the healthier bees. And that's a big problem for the colony, because in the end, the colony just can't keep up. It can't keep replacing them fast enough. The project that's most related to this stress is run by Dr. Mohammed Al-Baraki, who's in the laboratory, new scientist. He's interested in the sort of the impacts of this stress on queen honeybees and also workers, and has a new program just started on that. So some year you'll have to get him to talk about that, because it's kind of tackling these questions of how the colony survives these threats. Another big issue for bees, and as with us, is food. So if colonies aren't getting the right foods or a diverse food source, we think that too can kind of tip them over the balance, so they aren't as healthy, and eventually the colony dies. And so this is one of a number of studies that Dr. Miguel Corona in the laboratory is working on, and it's looking at bees and beehives that are moved around to different crops. Around the country, they go to different places to pollinate this food. And in doing that, they get a variety of diets. Sometimes they may feed on the same plant for a month, and that's all they get. Other times they end up, getting kind of a salad of different plants. And what they found in this study is that disease levels, and you can think of it for yourself, and if you're a student, maybe your parents are telling you this, if you eat plain bread every day and not as many vitamins, you're putting your body under some stress, and you might not be able to fend off a disease if it came your way as well. Honeybees do the same. The orange bars here, P, means it's a variable diet. There's like a lot of flowers in that food that they're eating. And the M bees ate a single type of flower. That was their only food source. And those M bees ended up getting sicker than the ones that had a diverse diet. So we're trying to figure out, and maybe the logical thing is to just plant different types of plants. But if you can't do that, you could probably feed those bees something that would diversify their diet. And that's where Miguel's going with this, is he wants to get kind of a super smoothie for bees, and that smoothie will have more of a more balanced diet. The other big challenge with bees is disease, and Sam Aban will talk about this in a minute, but I just wanted to highlight, some of the challenges that we see and that are facing bees. In this case, this is a virus that's all over the country. You've probably heard of viruses. We're tired of viruses, of course, and we're doing our best to avoid them ourselves. But this virus is all over the beehives right now, and it's a big deal for their health. What's interesting is that it's being joined by a sort of a sister virus lately that is also not so great for bees and has become widespread on the colony. And so back to kind of data and what we do as researchers, we wanted to know what's going on. How long has this other virus been there? Because we weren't really checking for it before. We didn't think we had it. And it turns out it's been now it's been 10 years that this new virus has been on the scene. And now that we know it's there, we can start to look at ways to manage it. One way is to control these mites that you'll hear about in a bit, because they carry this virus from bee to bee. And so that's a big way to do that. And then the other way is to look for treatments against viruses. And I wanted to show from my group. Again, this is last summer. Sadly, we were very quiet this summer. We didn't get into the lab, and we all missed it because it's fun, and we actually make progress when we're all in there. But last summer we had these studies going where we tested products that might control the virus. So we're testing things that are in plants generally that might be good for bees, and others. And so this was a really fun project. It had 6 or 7 of us. Actually, the last few summers we've had 6 or 7 of us between students and the full-time scientists in the lab, just trying to find a new treatment for these viruses. And we're super excited about this project. This is how we do it. We actually put bees in these little hotels, or not even hotels, they're pretty small. And we treat them. The bees actually come to us from our colonies with the virus. So that part's taken care of. And we just give them the medicine to try to cure them, to remove this virus. But we have to do it in these small cups, because we can't fit 100 beehives in the building and measure them effectively. So that's one element of our project. And I just wanted to show you the bigger picture, is that we're looking at how these challenges interact with each other. What's the outcome if you have this virus and the same mite at the same time? How does that affect the colony? And they really do interact. So it's okay, a bee might handle one stress, but if it gets 2, they're in more trouble. And on the mite side, this Dr. Ramsey will give you, he is the world's expert on these mites and really developed a lot of the thinking on their biology lately. But they are tiny, right? They're the size of, well, a little bigger than the head of a pin. But for a bee, they're huge. They're crawling around them and they're doing really bad things to them. So one of our big efforts right now is to find new treatments, chemicals, anything that's sort of effective to knock down this mite. And this is how you do that as a design. On the left, you have a bunch of these mites in a jar, and you basically just kind of expose them to different chemicals and see which ones really affect them. At exactly the same time, though, you want to do that to honeybees, because we don't want to invest in anything that might hurt the honeybees, let alone hurt us. So we're very careful to make sure we know that they don't affect anything besides the mite. And this is, with any science, you have to know where your results are going to go. In our case, we're really interested in things that'll be a long-term help for the bees. And to get them there, we'll actually have to get them through a process where they're regulated and actually labeled, and a beekeeper can go and buy these. So right, they're just available to the beekeeping population, and that's a big focus right now, is to get these medicines all the way through so that a beekeeper can actually use them. And now, since I noticed I don't think he had any pictures of himself in his own presentation, I will introduce you to Samuel Aban, who is running our bee disease diagnostic service. And here's Sam at his desk, maybe a couple of years ago, and then a little more recently, showing a group of scientists how to diagnose disease in honeybees. So a part of this job is kind of explaining to beekeepers and scientists how to, you know, how he works, how he finds out these sort of bad things in honeybee hives. And that's, of course, the first step towards curing the disease is to be able to find them in your colonies. So I'm going to, go back out, and it's all you, Sam. So I think you're muted, Sam.
Yes. Hi. Thank you, Jay. Okay, let me see if I can try to share my own screen and talk about the diagnostic lab. Okay. All right. Can you hear me, Jay, and everybody else? Great. Yeah, as Jay just alluded to the fact that, you know, bees are, you know, challenged with pests and diseases, and we sit on the front line of, you know, beekeepers trying to figure out what's going on with their colonies and also what can be done to it. As Ruby alluded to the fact that the lab itself has been around for a long time, over 100 years. And that's owing to the fact that since inception, the bee disease, the bee research lab was in the forefront in cutting-edge technology and also finding a ways to identify pests and disease in honeybee colonies. And one thing they realized was, yeah, research takes a long time. So how can we bridge this gap between research and service, finding a way to give beekeepers more information where they can use that information to actually manage their colony? So that's where the diagnostic lab or diagnostic service comes in. So we essentially takes what the researchers already know and try to turn it to diagnostics kits or diagnostics models to figure out what's going on with a hive, where the bees themselves, or actually the comb where the bee rear the colony where they raise the bees, and so they can be challenged with diseases and pests. So for us at the lab, you know, we get students also, you know, come in and help us in processing some of the samples that we receive. So you can see on my right here, there's typical samples that we receive in the lab year after year. So we can get samples on the top. That's containers of dead bees that were sent in from beekeepers, and is sent in preserving alcohol. And on the bottom of that is a comb sample, and it doesn't look like much, but that's actually a disease comb, and that's kind of a brood disease on that sample right there. On average, we receive about 2,000 samples a year that we process. And last year, we received about 2,500 samples. Most of the samples are sent in by actually the beekeepers themselves, who are figuring out that their colonies either died or dying, and they want to know what's going on with the colony. So a lot of times we get samples, either the bees the beekeeper collect, or the comb samples that maybe have disease on them. States also have inspectors that actually can inspect colonies, and they can collect samples of bees or the comb and send it to us for analysis as well. And now anyone can send in samples. It doesn't have to be the inspector, the beekeepers. Anyone can submit their samples, and once we diagnose, we send it back to the beekeepers or to the inspector that submit the samples. Now, and for the most part, we separate what we do from the adult bees and the comb sample. So for the adult bees, we examine them for one of the mites that Jade alluded to and in his presentation. That's a varroa mite. It's one of the devastating mites that affect honeybee health. And also there's another internal mite that's called honeybee trachea mite. So that one is a less problematic at this time, but also it can also create havoc in honeybee colonies. The adult bees themselves can also be challenged with disease, and one of them is actually Nuzima disease. Now, looking at on my right here, that's the varroa mite. It's varroa destructor. So it's a external parasitic mite, and I'm sure Dr. Sam Ramsey, who's a foremost expert in that field, will discuss further on the destructive nature of this mite. So it presents a threat to honeybee colony, and it's active transmitter of viruses. And so you have the combination of actually the bees the mites feeding on the adult bees themselves, reducing their lifespan. Also, the bees don't die from the actual bees, the mite activity. They can die from the viruses those mites actually transmit. Another mite also is the honeybee trachea mite. So this one is an internal mite. So like I alluded earlier on, it's less problematic, but some of the chemicals that are used to treat the external varroa mite also act on this internal mite, and so it becomes less and less of a problem. So another disease that affects the adult bees is actually Nuzima disease. So it's a microsporidium. So it's actually affecting the gut of the honeybees, and also reduce lifespan, and actually kills eventually it kills the adult bees. So there's two species of this disease actually. So it's Nuzima apis and Nuzima serena. But for the purpose of this presentation, it doesn't really matter, because our diagnosis, we try just to detect the disease itself and not so much what type of species is present, because the actual treatment is the same whether you have the apis or serena. Just give you just a background of what we're seeing in the lab in terms of varroa mite prevalence. And we see it from last year, about 36% of the adult bees that we examine, we find varroa mite on them. On average, we get about 11.4 mites per 100 bees. Now, it may not sound as a big number, but anything below 5 mites is good, and anything above the level of 5 mites per 100 bee, it's pretty much like a death sentence for the colony. And that's owing to the fact that the mites replicate very rapidly. And so they can go into the brood cells and they can multiply 8 times versus one adult bee that can develop from that same cell. So you can see if you have, thousands and thousands of cells and mites infesting those cells, the bees doesn't have any chance to survive. So on average, we can find that the maximum is about 137 mites per 100 bees. And so definitely, if you find that kind of infestation level, definitely that colony is dead. And in our experience, most of the time when we find that we check back with the beekeeper, most of the time those colonies already dead. So that's just to confirm that that the mites actually play a great part in the demise of that colony. In terms of Neisseria disease, again, also, so 35% of the colonies, that is the sample, we can find, Neisseria disease. The spore ranges between 50,000 to 129 million spores per bee. Again, Neisseria is not much such a death sentence, but if left untreated, can really cause a very difficult time for the colony to survive, because now the bee is going to be weakened, and then if they get any surge in mite level, they can really destroy the colony. We also find about 59% of the sample have over 1 million spores per bee. It sounds as a low level, but during the winter time, those can rapidly increase, and then the bees actually were not able to rebuild their population quickly. And if that's the case, then the colony will, die out. And so, Jay showed a graph about winter losses, and in many cases, the Neisseria disease also play a big part in bees dying when they overwinter. I'm going to switch off to the brood disease. And so this area, American firebrood, which is one of the devastating and destructive of the brood diseases that we diagnose in a lab, and followed by European firebrood. And at a lesser extent, we get chalk brood, and also the virus, which is sac brood. That's less and less of a problem, but we do from time to time diagnose colonies with that disease. Now, in terms of American firebrood, it's a spore-forming bacterium. And now, if you imagine have one cell can have over 2.5 billion, spores in one cell, and so it can actually transmit rapidly in the colony and kill the colony if left unchecked. It's highly contagious and usually kill the colony, and the bees would just dwindle, and eventually they would die out. And last year, we about 12% of the sample diagnosed with American firebrood. In terms of European firebrood, it's a non-sporing bacterium, usually caused by stress of the colony, and also does not kill the colony, but the reducing population and the stress that is brought about to the colony will weaken the colony, and eventually other opportunistic bacteria and pests will come along and finish off the colonies. And so in most cases, we saw opportunistic organism also will flourish in that environment where European firebrood is already established. Just an example, last year also we find about 24% of all samples were diagnosed with this disease in our lab. And in terms of diagnosis, I now just talk about all the stuff that we see in terms of disease and pests, but the actual work is done by using I would say my best tool, which is the microscope. And so we just collect a sample We fix it to a slide, and then we look it under the microscope. Now, what we're looking for is just pretty much the distinction between the bacteriums that are present. So in terms of the American firebrood, which is the Bacillus lava, which is a causative organism, we look at the shape, and we look also to see if this is a Brownian movement. And that's very unique with this organism, as opposed to the European firebrood, which is caused by Melissococcus plutonius. It tends to be fixed and not moving as much, and they generally be found in pairs. And so we use those differentiative markers to actually distinguish which of the diseases that are present in any given sample. And now, for us, we're excited to actually find something in the comb, but for the beekeepers and inspectors, that's really bad news when we send them a report and saying that you have, especially for American firebrood, because in generally, there's no actual treatment that can actually combat the disease. So in most cases, they recommend that, you know, the beekeepers have to destroy the whole colony. So, and that's actually having an economic impact on the beekeepers as well. So we don't take that lightly when we make this kind of diagnosis. So there's other steps that we take to ensure that our diagnosis is confirmed, and so that, you know, the state inspectors are making the right call in the colonies. And so just looking under the microscope, this is what we actually will see to make the determination what actual organism is present. So my left hand side here will be on your right. That's the Penibacillus lava on the microscope. And on the right will be on your left side, that will be Melissococcus plutonius. And you can see it in general cluster, but the sizes are they varies. And so we take, you know, advantage of the variance in size and the morphometrics on those organisms to make a determination. And also we can take a further step, and actually we can culture this organism once we make a determination which one is present. So in the case of the American firebrood, we can actually culture them. It takes about 3 days, just pure culture media. And you can see to my left, which should be probably on your right, that's the inhibition zone is shown right there with the arrow goes through across that zone. So, and on my right will be on your left side. As you can see the measurement there are very different. And so on my left will be on your right. There's a colony that the actual antibiotics is actually is working. And so if they treat that colony, they better will have a good chance of containing the spread from the adult bees, because the actual disease doesn't affect the adult bees. On the other hand, the other colony we have a harder time, because that is showing a resistance. And so you can see the zone of inhibition right there is very small compared to your right, where the zone right there is a bigger zone. So that's another way we confirm and also help the beekeepers understand what's going on with their colonies, and also inspectors to take appropriate actions. And so in terms of the numbers last year, we found about 70% of the sample was susceptible to the antibiotics, and 24% were resistant. And so the most commonly widely used antibiotic is oxytetracycline. And so that's the one we actually beginning to see some resistance build up. And the other antibiotic is tylosin, which we haven't been seeing any resistance building up yet on that. So just to sum it up, just give some few points here that firebrood disease and varroa mites, pose the most significant threats to honeybee health. And also nationwide, we're seeing some resistance to antibiotics and miticides. And Jay also alluded to that, the difficulty in actually controlling varroa mites, and the longer the use of antibiotic or miticides, they build up resistance. And fibers is most destructive, like I alluded to earlier on, and being owing to the fact that it's spore forming, and also it can be transmitted, by beekeeper themselves through, beekeeping activity, and also the actually nursing bees can transmit the disease while doing feeding and nursing, caring for young emerging brood. And finally, varroa mite is destructive to both the brood and the adult bees. And so you see varroa mite is pretty much in the center of the beekeeping world lately, and researchers, scientists all over are trying to figure ways to control this mite destructive activities. And so I will stop here now. Hopefully we have Dr. Ramsey that can illuminate further into the mite world for us.
All right. Thank you, Sam. Yeah, I think Dr. Ramsey is on. There we go.
Okay.
Hi, everybody. So my name is Samuel Ramsey, and has already been discussed. I'm a researcher with the United States Department of Agriculture, but I've also been conducting some of my work in collaboration with the university, Chiang Mai University in Thailand, and some work with the Van Ingelsdorp lab at the University of Maryland. And so I'd like to just tell you a little bit about the work that I've been conducting with our partnerships at the USDA. So this presentation is about the mites that you have already heard a bit about. They've been mentioned from what Dr. Jay Evans discussed. They've been mentioned from what Sam Aban was discussing. And I'm going to give you a little bit more detail about how such a tiny creature can cause such a big problem for honeybees. So the despicable mites. Let's see. Good. So honeybee colonies are these robust organisms. We call them superorganisms because none of those bees is capable of existing without all of the rest of those bees together. They form a little conglomerate that keeps everybody healthy and happy. So all of those bees, each one is like a cell in your body. And by getting a bunch of the cells sick, you can get the whole body sick. When those bees want to grow and develop, this whole colony is structured based on their wax. They produce this wax, they build cells out of it, and they can store honey inside of those cells, and they can store their offspring inside of those cells. And so when their babies are developing in these cells, this is where we see a lot of the problems, because their babies are these big sacks full of food. They are nutrients inside of a small, thin skin. And there are a lot of other organisms that really want to eat them. The bees work hard to keep them out of the colonies, but there are a couple of mites that have done a great job of still getting inside. One of those mites is Varroa destructor. And if you didn't notice, you are currently staring into the face of evil. Every beekeeper on this planet pretty much has had to deal with these creatures, except on a few islands and on the continent of Australia. They've made their way all around the world, and everywhere they've gone, they've destroyed tons and tons of honeybee colonies. So this is a close-up of the face of evil, but if you're having a difficult time locating that face, I can help you out just a little bit. If you'll look between those two big front legs, that is the face of evil. That face has no eyes. It's very difficult to locate even where the mouth is. There's no nose, but it's a very strange organism. I'm sure that that's something that it conveys to you in that. They first reached the United States in 1987. Prior to this point, they were in Southeast Asia, and they had started to spread around the world. There was, up until that point, there was only one section of the world that they'd really been in, and it was just Indonesia, Thailand, Malaysia. That was their original range, and they only fed on Asian honeybees. But then at some point, a really enterprising mite decided, I want to see the world. And this mite transitioned from her connection with the Asian honeybees that kind of had a lot of ways of fighting off these mites to Apis mellifera, the honeybee that is spread all over the world, and we call it the European honeybee. And by doing this, they found a host that was a lot easier to exploit, a lot easier to attack, and that allowed them to move pretty much everywhere. So the only places where you really don't see them now are Australia. The reason why I show you this map is because we learned something really interesting recently. We found that as we conduct research, it can be really difficult if we wait until the parasite arrives to really start intensively conducting research. And so you've probably heard about things like the Asian giant hornets that have recently been dubbed murder hornets. When creatures like this get into the US, it's really helpful if we have some research that we've conducted about them, information that we know about them, so that we can use that information to stop the creatures from spreading and to stop them from becoming established. And sometimes, even if we have tons of information, we still can't stop them. Well, when we were trying with Varroa, one of the issues that we had is that a lot of the papers that were written about them were written in China and in Russia, because these were the first two areas where the parasite became a big problem. Prior to that, they were only in Southeast Asia and only on Asian honeybees, and so they weren't causing a lot of issues. But that meant that a lot of the papers that we would have needed to use to learn about these parasites were written in Russian and in Chinese, and that made it a lot more difficult to get the information that we needed to understand these parasites. So fast forward many, many decades, now it is, now we're in the 2000s trying to understand this parasite, and there's a lot about it that we're realizing that we can still learn about it in order to control it. But there's another parasite as well, and it's the Tropolelaps parasite. And this one, similar to Varroa, spends a lot of its time feeding on the baby bees. It goes inside of the cell, it pushes its mouth parts through the bees, and causes a lot of damage to them as they develop. So this is what a larval bee looks like. This is the sac of nutrients that I was telling you guys about. And this is the pupa that it eventually turns into before it turns into an adult. And the parasites get inside of the cell and start feeding on this creature when it is at its most vulnerable. It can't fly away, it can't fight back, it can't bite anything, it doesn't even have those sorts of mouth parts at this time, and so it's just a squishy, soft creature that can't move. These parasites came from a really big, a much bigger honeybee species, the giant honeybee. And because they've made this transition from a larger honeybee to a smaller honeybee, but not a lot has changed in that process, they're still feeding on a large amount of tissue from a smaller bee. And that's a big problem because the amount of damage that they cause to our honeybees is pretty substantial, because they're feeding on them the same way that they fed on these giant honeybees. You also might notice that there's a very similar map here. So this is the distribution of the Tropolelaps mite currently, and this is the distribution of the Varroa mite currently. But Tropolelaps started in Southeast Asia and then started moving outside of Southeast Asia to China and India and South Korea in the same pattern that Varroa did. And so it's important that we keep an eye on this parasite too, just like with Varroa. It's very important for us to know more about it and to keep a really good eye on it. So I've been working on doing the same thing because I've been very concerned about how parasites can spread from just one introduction. There's a really interesting story that I was hearing about in one of our international meetings from a friend of mine who works in Reunion Island. And he's their bee inspector. So he goes around and he does a lot of what Samuel Laban was talking about. He makes sure that the bees don't have foul brood or any of the other really big concerning diseases. And he found that there was a new parasite on their island. They didn't have any Varroa mites and everything was going great on their island. But unfortunately, as the result of just one introduction, so just one or a few mites all came in one area on this island, and in just a few months, in less than 5 months, they had spread to most of the colonies on Reunion Island and continued to spread out from there. Now they're present in the vast majority of the colonies there, and they have raised the rate of death in the colonies from just a couple percentage, just when it was around just 1 or 2%, and now it's up to 40% each year, and it's very concerning for them. So We're trying to avoid the same sorts of things. And the ways that we can avoid that are through knowledge, by better understanding the parasites and their host. So when I was an undergraduate, I worked on understanding how this parasite works. This is Varroa destructor, and it's not immediately evident what Varroa is doing to that bee. Is it feeding? Are its mouth parts in there? And if it's feeding, what is it feeding on? Well, in order to understand that, I had to figure out what is it that's under these plates on the bee's body, because this is the only area where we ever found the mites feeding. And so it became very clear to me that the mites are really interested in something that's in between these plates on the bee's body. It turns out, using the electron microscopes that are present at the USDA and our connections with the electron microscopy lab, we did a lot of work just cutting through mites and bees at the same time and actually looking at what we could find. And this image is an electron micrograph to cut directly through one of the mites that's currently parasitizing one of the bees. You can see inside of its body, and you can see on the other side of the membrane where the mite is feeding, that there's this abundance of tissue called the fat body. And it's actually the bee's liver. And prior to this point, we were under the impression that this parasite was a lot like a tick, that it was removing a little bit of the bee's blood. And it was really confusing to the scientific community. How exactly is this parasite killing bees so quickly by only removing a little bit of its blood? For us as humans, if a big tick or a big mosquito landed on us and took out a bunch of our blood, it would be a problem if they took a big portion of it. But we can generate more blood and things, and it's as long as these creatures aren't generating huge volumes of viruses into our bodies, then we should be fine. And while we know that Varroa has viruses, it seems like they're capable of causing a lot of problems that aren't related to viruses at all. What we found is that they were breaking down the bee's liver into a sludge and ingesting that. And without the liver, the bees have a really difficult time getting through life. One of the ways that we figured this out was actually by coloring the bee's liver the color red using a fluorescent glowing dye, and then dissecting the mites. I dissected the mites, and we could open them up and look at their digestive system and find all of this red glowing tissue that we knew came from the mites' liver and not their blood, because the chemical that we used to dye it was hydrophobic. And so that kept all of the chemical in the very fatty liver tissue, which is called the fat body in bees, and not in their blood. So this was really a very important thing for us to understand, because the liver of the bees, it relates to all of these different functions in the bee's body. It synthesizes their proteins and their fats, it breaks down pesticides, it's very important for their immune function, and without it, the bees get very sick and they don't do very well. But what we've also found is that with this other parasite that we were discussing, the Tropolelaps mite, it can cause a lot of issues that are similar to the Varroa mite. And so I actually went to Thailand to better understand how that parasite works. And I'm still working on that data now, but some things that we do know is that the mite actually bites a bunch of holes into the bee's body, and each of these holes causes a wound that scars and doesn't allow for that section of the bee's body to move correctly anymore. It causes a lot of scar tissue. And so when they bite areas of the bee's body when they're developing, if that section was to be the bee's legs, that section of their leg wouldn't work properly anymore. It would be permanently bent in one angle, and the bees wouldn't be able to walk properly. This kind of damage and a number of other elements of damage from viruses and a breakdown of tissue can cause the whole colony to fall apart. So what I wanted to understand is what happens when the parasite is present inside of the colony, inside of those sealed cells. It's very difficult for us to see inside of the cells. We don't know exactly what's going on when those cells are sealed up. And so we created this system that we've been calling Mite Insight, where we can look through a glass wax cell. So half of the cell is wax, half of the cell is glass, and we can actually see the parasitic mites. This one is Tropolelaps, this one is Varroa. We can determine how they interact with each other, how they interact with the bee brood, and we can learn a lot about them that has been very difficult for us to learn in the past. We've been working really hard to do this to avoid to avoid ending up in a situation where we don't understand these parasites when they arrive. So in Thailand, we were even digging up colonies, pulling them out of the ground because there are a lot of natural colonies in the ground, and looking at how under natural circumstances, how do these Asian bees control parasites like Tropicolaps and Varroa? And by doing this, we have learned a lot of information that has been very helpful to understand, and that we are disseminating to the beekeeping community so that we can learn as much as possible and use that information to help as many people as possible. So this is me trying to get all of that work done in just a few months. This isn't sped up, by the way. This is just how fast I had to work to get all of this done. I'm kidding. It, of course, is sped up. I'm going to be continuing this work hopefully next year when the pandemic is over. And I would like to thank all of you for listening. And I think from there, I can turn it back over to Jay. Let me see.
Yeah, or to our hosts. I know there are some good questions that came in, but I also imagine people might have specific questions about how you work on these crazy mites and how you fit your video camera into this tiny hole. So, what do you think, Julio or Ruby? Should we tackle a couple of these questions?
Yes, thank you. I just apologize. I was trying to get myself off of mute, but we do have a few questions that came in, so we'll turn it over to our chat box. And so our first question is, did each of you always know you wanted to study bees?
That is a good question. I did not. I thought I'd be a marine biologist or some, other branch of biology, and I found a passion for ants. So turns out that ant colonies are an easy inroad to studying bees. So I came in through the ant route. But I guess I did pretty early on, and I know from Dr. Ramsey's background especially that we shared an early interest in sort of natural history and what's going on around us in the environment, and so that was there. But I had no idea what I would do as a grown-up, and I very luckily found a job doing something I really enjoy. But there are lots of branches of science that I would enjoy, but I think bees are pretty cool.
Yeah, hi. On the other hand, I mean, I never dream about working with insects in general. I think my experience or encounter with bees would be just, what I suppose was a bee that stung me one day. I was on a bus stop and I was like, oh, you got stung. I mean, in general, we used to say, something sting you, like, oh, it's a bee. And I know maybe that wasn't the case, but yeah, I mean, I was interested in chemicals, chemical reaction, biological matters and stuff like that. And maybe for you guys listening, students, and you never know where the road takes. I started on the lab just as an intern myself, and I just fall in love with working in insects. So when I showed up, and my boss then was asking me, "You have any interest in bees?" Like, "Are you sure you want to work with bees?" It's like, I'm not sure, but I gave it a try because it was interesting for me because my background also translates to what was going on in the lab at the time. I was also able to mix chemicals and stuff like that, and bees are biological matter as also an interest to me. So, I started growing up my skills that way, and here I am. So, yeah, I don't know what you think, Dr. Ramsey.
Oh, well, I can tell you this is my dream job. To actually have someone employ me to learn about insects is what I dreamed I would be able to do since I was a little kid. So when I was 7, I told my parents, I want to be an entomologist when I grow up. And I went through school thinking about all the different ways that you can be an entomologist. But the one that ended up really catching my interest in graduate school was being a researcher of bees. And one of the reasons for that is because I love the idea of symbiosis. I think it's really fascinating when two creatures have this weird dependent relationship on each other where one of them needs the other or both of them need each other. So the mutualisms, the parasitisms, even really close relationships that are predatory, I've always found them really interesting. Honeybees have some of the most fascinating mutualisms in the world. They have a mutualism with human beings where we're both benefiting from each other, where we're protecting the bees and we're getting honey and things from them. They have a mutualism with flowers. That's one of the oldest ones in all of existence, and it's really fascinating. But they also have some weird parasitic relationships with these mites. And since I've always been fascinated by mutualism, and I realized that people could even use this sort of interest to help other people. So when I realized that I could help by kind of dedicating my interest to the study of bees, I found it really exciting, and I've never really looked back.
Thank you. Our next question, kind of along the lines, is what are your recommendations for students who may want to study bees or any other areas of research? Any lessons learned?
Wow, that's a good question. I think it more depends on what type of bee research you want to do. As you know, we have chemists, microbiologists, people studying behaviors, genetics. So that would affect, say a summer job search as much as anything. Although it is worthwhile certainly to say if you're approaching a professor or a scientist here at the USDA to say that you're very interested in bees and working with bees, but also to kind of look at their projects. I've always found it's good if you're going to write to, inquire about a job, kind of see what they're doing. If they're doing genetics, make sure that's something you'd want to do. Because a lot of us, I got into it because I loved swinging a net outside and walking outside, but my lab currently is kind of an indoor lab, so we do a lot of genetics, and so I feel badly hiring someone who knew they wanted to be outside the whole summer. So you kind of do that bit just to figure out the type of science within bees. And then my, I guess, main advice is be pretty relentless about jobs. There's another question about whether you get paid or not, and you do. Yeah, summer jobs, you should be paid an hourly, a stipend or a wage. But you might have to, it might be like other fields where you have to approach 10 people to find an opening where they have the money, the funding and that. So yeah, doesn't hurt to, have in sort of January or February, have your CV in shape and start marketing yourself. And that's the way to get in the door. And that's true for any field, but it's really true for science.
I'd also say passion is a really good way to get your foot in the door. When you can convey to people that they're that you're very interested, very excited about what it is that you study, it's kind of a contagious thing. People like to see it. They like to believe that that this isn't just something that you're doing for money, but it's something that you're doing because you're passionate about the subject, you're excited about it, and so trying to convey that can be really helpful. Showing that from the things that you've decided to do with your time in the past, being able to show that from undertakings that you've worked with as an undergraduate or as a high school student, that helped me a lot with getting my foot in the door to different places. So never underestimate the power of passion.
Dr. Aban, you're on mute.
Happens to us all.
Yeah, I was just saying, I'm really gonna second that what Dr. Ramsey just said about having a passion, have a drive, and also just having an open mind and what you want to do. As students, we already have the set goals and ideas what our future is gonna look like, or future career is gonna look like, and whatnot. But if you really think about it, you get educated to absorb new ideas, open yourself to new thinking and be creative. And it's just like the same way going out there and finding a job or looking at potential opportunity for a job. If you're not open-minded, then you limit yourself to just um, just what you believe or aspire to, and not in your horizon or what you can be. And I can say that for myself too. I was dead set to saying, "Oh, I want to be a biochemist." Because I love doing these things, and then I was like, maybe I give it a try. And so my open-mindedness, if I can say that, it led me to where I am today. And I'm sure for a lot of students, I'm sure they're kind of thinking about these things, like what they want to be, what they want to do, and some may have a set goals, they want to reach, a set career they aspire to be. But also, I would just say, if you have an open mind, that can lead you to that dream job, you can love doing and be excited to wake up to go to every day and be a part of a community that actually, doing something productive in society. So just having an open mind and what you want to do in the future. And by the way, don't be afraid to reach out, and to other, people in the field that you don't think that you're interested in. Like entomologists. I frankly, I never thought about what is entomology. Sometimes maybe you can, for the research yourself, like try to get an understanding of, all the fields that actually knew about that are up in the headlines. That being a scientist, because I would say first things that would come to mind are scientists, being a researcher, be a doctor, all of the medical area, and entomology would probably be on the lowest tier of what students are thinking about.
Thank you. One other question that we had is very cool research. Is there a way that students can participate or connect with your ongoing research?
I think, yeah. We do have a website at the lab that has the different projects. I mentioned the nutrition project and the queen and colony stress. And, yeah, email is probably as good as anything to reach out to one of the scientists. You can certainly email to me as well, and I could try to direct it if it's a better fit for someone else in the group. I guess, yeah, currently, given that we did sometimes have informal open houses, things like that, and that doesn't seem to be on the schedule currently, sadly. So I would say the best is to reach out by email and, and again, check and see which projects look especially interesting to you first. And we'd be happy to hear from people. So.
Absolutely. One of my favorite parts of being a researcher is having the opportunity to teach and mentor. And when you're working, if you're working in academia, you're teaching students on a regular basis, but you also get the opportunity to teach if you work in the government, simply by teaching individuals that you work with. Sometimes we'll have interns or other people, and everything that you're doing is a learning experience in one way or another. And so if you happen to have a passion for one of the subjects that you've heard about, reaching out, if there is some way that we can be, that we can help bring you into that process, we'll certainly try. Right now with the pandemic, it might be a little bit more difficult than normal, might be a little complicated, a little bit, but still worth reaching out.
Yeah.
Here's one for.
I'm sorry, actually, I was on mute. I apologize. Is it the last question I was asking, or next last question was, are there any USDA programs that are available to help pay for students to study bees?
Yeah, and I think again, it would be through the center, whether it's here or maybe a university lab. You really have to get the ear of the scientists there, and then they can work with you to try to find the funding and apply themselves for funding. So, but that's there are paid opportunities for summer students. So again, as the people, as Sammy and other mentioned, it's you gotta really push it, get out there early.
Mhm.
Thank you. And the last question, I guess I'll ask for each of our presenters today is, what made you decide on USDA, working at USDA, and what might be next in your career journey?
Guys.
Mm.
I'll say my career journey is kind of not quite going on the downhill slope yet, but I'm probably here for my career. Yeah, so I decided 20 some years ago to come to Beltsville, and I've been here ever since. And I just enjoy the people, as Sam mentioned. Working with beekeepers, you always have this kind of, they don't always agree with us, but they're a fan club, and they have needs. So one of the neat things, and it's probably true for other agricultural research, but I don't have experience, but yeah, we go talk to bee clubs from 10 people in a suburb somewhere to the state meetings and international meetings. So there's a big community of beekeepers, and that's really fun, just to see what they're concerned about and to talk a little science with them. But yeah, so I guess these two, not to put them on the spot or anything, but they may have more to say about where this could lead to. But for me, it's hold steady here. I'm enjoying the ride.
Well, Jay's being a little bit modest. I mean, he's reached the top of the bee lab. He's the guy that runs things. And so if my career were to go in a direction where I become the Jay one day, I would not be upset. I'd be pretty happy with that trajectory. But in general, I am still at a point now where I'm not entirely sure where exactly my career is going to take me. And that's one of the most exciting and for some people, sometimes the most frightening parts of the experience. I'm only a couple of years out of graduate school, and so I'm still sort of doing the dance of the postdoc, of figuring out where do I go from here? Where can I do the greatest good? Where can I actually make a living and enjoy my life? And so Sam was discussing earlier how important it is to keep an open mind about options. And I think it's very important that throughout your career, however far you go, wherever you end up, that you continuously keep an open mind, because there are incredible opportunities that will present themselves to you. And if there's only one thing that you're singularly focused on doing, like one direction that you could possibly go with your talents or your passions, you might miss out on another direction that those talents and passions can take you. And so as much as I want to be an entomologist, there are so many different kinds of entomology out there. Originally, I was just fascinated by praying mantises. I thought I was going to be a guy who only studied the praying mantis. And oddly enough, things came around to me being a bee guy and studying parasites of bees. And so maintain an open mind. I'm thinking that as long as I have the opportunity to both conduct research and teach, like interact with via mentoring or through a class students, I will be very happy with the direction that my career takes.
And yeah, I mean, I'm sort of trying to chase Jay and Sam. You see Sam, put a very passionate about speaking and engaging, and and it's quite a rare opportunity you have in our field to interact with people, and also not just just a selective being either scientists or just ordinary beekeepers, and it doesn't come along in other profession like we have here that, we can go out there, speak to, to their concern, absorb it, and also trying to find a solution to those problems they present us. And so, it kind of in a way, keeps you going. And because we not only thinking about what we do, we're thinking about the beekeepers, what they're going through in terms of how they're going to manage a colony. And for us, it's how are we going to find those solutions to help those beekeepers manage a colony. And so, there's a lot of people that spend a lifetime, and suddenly we have spent good part of our life doing that as well. So, and we also, building up the way for, others to come along and build upon the knowledge that we already generate and constantly producing. And so it's very important for us to, just keep the drive going and be open-minded. I know, yes, a lot of opportunities that are available, and so, but we just want to leave the mark where we are right now. And so others might come and, take that point and hopefully find the solutions that, elude us so far, because, who am I? That's going to take a lifetime to figure out what the heck is going on with this mind and all these tricks is pulling on us, and so how are we going to contain it? And so for me is give me the, the hope that, there's something there for me that keep me interested, there's more to, discover. And so, yeah, I'm kind of in a way settled in where I am right now and just focusing on, trying to build more knowledge, career-wise, build up myself, and also just focusing in ways to help, the beekeepers and the bee community as well as best as I can.
Awesome. So this concludes our August installment of Faces of Agriculture, the USDA Speaker Series. We would like to once again extend a thank you to both our presenters and attendees for taking the time to learn and grow with us today. We invite all of our attendees to visit our USDA Youth and Agriculture website at www.usda.gov/youth to learn more about the opportunities to engage and empower the next generation of agricultural leaders. We look forward to having you join us again for next month's installment of the Faces of Agriculture. Thank you.